All right, kiddos, so today we're going to discuss transcription, but I wanted to show you first the difference really between replication and transcription. And the reason I want to show or compare them really is because they both involve the copying of a DNA molecule, but for two different reasons. So DNA replication, which is what you wrote your last CRQ on, in DNA replication, the whole purpose of that is to make an exact copy of the entire DNA molecule. So all the DNA, in your cells, in the nucleus of your cells, the purpose of DNA replication is to make copies of that, and so that when the cell goes through mitosis and then splits into two daughter cells, so that all the new cells have the exact copy of the original DNA code from the time, the organism was a zygote. Okay, so DNA replication is simply just making a copy of the original DNA code so that it can get passed on to new cells, so that they always have the same code. It only occurs in the nucleus. There's only DNA involved, no RNA, and like I said, it creates an exact copy of the entire DNA molecule to be passed on during mitosis. Transcription, though, is also a process that makes a copy of DNA, but it only makes copies of segments of DNA so that proteins can get made. And remember, we need proteins to be alive. And instead of making another DNA molecule, what happens is mRNA, messenger RNA, is the copy of the segment of DNA. So just a piece, a section of the DNA gets copied, and that copy is messenger RNA. It's made in the nucleus, but RNA is able to actually leave the nucleus. Remember, DNA can't leave the nucleus, but RNA, once the copy of the segment of the DNA molecule is made, the RNA can leave the nucleus, and then it travels to different organelles in the body or in the cells to tell those cells how to make proteins. That's a lesson for next time. But so right now we're just going to talk about transcription, which is the process of making a copy of just a segment of the DNA. And that copy, we call it messenger RNA. So the big picture, right? We've read this before. In order to be alive, your body has to make certain proteins. The instructions on how to make the proteins are held in your DNA. RNA deciphers the code and delivers the instructions to the ribosomes in the cell. The ribosomes then read the instructions and build the proteins. So DNA is the keeper of the code. RNA is the copy of the code, right? That reads and delivers the code to the ribosomes, typically where the proteins get made. So that's the big picture. So today we're going to discuss this section here on how RNA, messenger RNA specifically, is made, because messenger RNA is just a copy of a section of the DNA molecule. So in order to be alive, our body has to make proteins. The proteins are made in our cells. Our DNA holds all of the instructions for making those proteins, but we don't want to use up our DNA. We need to keep a copy of it, the original DNA code, in all of our cells inside the nucleus. So before a protein can get made, the cell makes a copy of the DNA segment and uses the copy to do the work. It's kind of like maybe your teacher in a brick and mortar school would have a copy, like the original copy of an assignment that you have to do, and then your teacher would go make photocopies to pass out to all the students. So it's the same idea. DNA is the original copy, and then RNA would be the photocopies that get passed out to all the ribosomes to do the work. So what now? Today, we're going to learn how the cell makes that copy of the segment of the DNA. The copy that gets passed out to all the ribosomes, right, in the cell to make those proteins, gets made in the nucleus. So the RNA gets made here in the nucleus, and then it migrates out into the cytoplasm of the cell, right? That's the fluid portion of the cell. And the RNA is then able to tell the organelles in the cell how to make the proteins that the body needs. But again, we're going to take care of that next week because that's a different process. So here is the official, step-by-step process of transcription. You might want to pause here for a second and take note of this, because I promise you, you're going to need this for later. But here we have segment, right? A section of DNA is used to make a molecule of mRNA. First thing that happens inside the nucleus, just like last time, DNA unwinds. The enzyme helicase comes in and unzips the DNA. RNA polymerase comes in and moves to a specific point on the DNA strand. The nucleotides come in and pair up, which begins the process of making mRNA. And after all the nucleotides are paired, mRNA moves out into the cytoplasm. So if I were you, I would pause here and maybe write down these steps, because I promise you're going to use them. You're going to use them a little bit later. Okay, so next, here we go. Before transcription, right, DNA unwinds. Here's a diagram of a DNA molecule in a eukaryotic cell. We show it untwisted for simplicity. You've seen this before, even though it would normally be twisted into a double helix with parts of it wound around histone proteins. Okay, so this is DNA molecule and it is shown untwisted. The DNA unzips, just like we learned in DNA replication. So far the steps are the same. The DNA unwinds, the DNA unzips. Remember, this is helicase, right, that comes in and unzips the DNA molecule. Now we're officially in transcription. So after the DNA unzips, this is kind of the official first step of the process of transcription. Something called RNA polymerase begins the process of transcription, and this something called RNA polymerase is an enzyme. So this is where transcription officially begins. RNA polymerase recognizes the sequence of nucleotides that needs to be copied and moves to that point on the DNA. Remember, we're not copying in the whole DNA molecule here. We're just copying sections of the DNA molecule. So the RNA polymerase is like, hey, I know where to go. It moves to the portion of the DNA molecule that needs to be copied. Then, right, the RNA nucleotides pair to their template. So this RNA polymerase, the enzyme, helps the base pairs pair up. So you have your original DNA molecule here on the left, also on the right, and then the RNA starts to get made. Okay, so this enzyme is like, ooh, this T, right, needs an A, and this C right here needs a G, and this G needs a C, and this T needs an A, and this C needs a G. So the enzyme, the RNA polymerase, brings in all the little bases and pairs them up. The only difference this time, though, is that whenever you see an A, right, on the original DNA molecule, if there's an A, instead of a thymine coming in, it's going to be uracil. Okay, if there's a T on the original DNA molecule, that's fine. It'll just bond to adenine. But if there's an A on the original DNA molecule, it's going to have to bond to what we call uracil. So this left side is the original strand of the DNA molecule, and then the mRNA is being made here on the inside. RNA nucleotides get all paired up. You can see them right here. So we have RNA made on this side. This is the original DNA strand. The mRNA strand leaves the nucleus. So here's the nuclear membrane, right? So the mRNA strand leaves the nucleus and travels into the cytoplasm of the cell and then goes off to the different parts of the cell to go ahead and make proteins. Again, that's for next week. Okay, so transcription makes a copy of a specific gene. The copy is then used as instructions to produce a specific protein. Transcription is the process that converts genes in DNA into portable blueprints or messenger RNA molecules. Each mRNA molecule carries a copy of the code for one gene. During transcription, the enzyme RNA polymerase brings in the correct nucleotides to build an RNA molecule. The same base pairing rules used in DNA replication are used in transcription, with the exception of uracil being used as a base instead of thymine. Once mRNA is complete, the DNA closes back up, and the mRNA carries its information to ribosomes. All of the types of RNA, mRNA, tRNA, and rRNA, are transcribed in this way. They are all transcribed in the nucleus, and they all migrate to the cytoplasm later.